Yes. So, good evening to everyone. So, welcome to the second session on internship and certificate course. Internship and certificate course on artificial intelligence and machine learning approaches on drug and vaccine design in cancer. So, do you remember what exactly we have seen in last session? So I think the slides are shared at present, and also I can show you the schedule. So we are discussing something about introduction to biological databases. Majorly, we are discussing about oncology databases, isn't it? Before that, I like to have an interaction. Like, is my voice audible to you? Is the internet strength is okay?
Yes,
Yes.
sir.
Okay.
Yes.
Thank you for this interaction, and I thank all the participants. To receive a feedback on yesterday's session that many are praised. Nearly more than 92% of participants has provided a good feedback on the session, on yesterday's session. I anticipate the same kind of feedback I can receive even in the other sessions, in the other 13 sessions. Because the last session is basically we have a test MCQ. Okay, in the second session, okay, is something related to target discovery, that is on target identification, modeling, and validation. What is target actually? We have two technical terms. One is ligand and receptor in computational biology, that is especially computational related drug design. We have two receptors. One is ligand, the other is receptor, isn't it? So if you see this uh, receptor, which is most important, we have to target for a specific, I think the participants can join earlier. So I just waited for 5 minutes. Don't make me wait for more than that, because even if you join earlier, it won't give disturbance to the others, isn't it? So try to join earlier, 5 minutes earlier. If the session is 7 o'clock, try to join at 6:55. Even myself, I join at 6:55. I sharply start at 7 o'clock and complete at 8 o'clock. Or you are losing some time session in joining in between, and we have some kind of a noisy disturbance. Okay. So next session onwards, kindly join on time, 5 minutes before. Okay, now I just take on this new chapter, that is on target identification, modeling, and validation. So initially, we have seen what are the databases present and what are the types of data present in the databases, isn't it? Now we'll be seeing about protein target receptors and what are the various databases available for downloading the 3D structure of proteins, which we call it as targets, which are we call it as receptors in computation aided drug design. Okay, so we'll be checking to download the 3D structure from the databases. If not available, we have to go for modeling. Okay, for unknown protein structures with a known protein sequence, we can able to go for modeling, and finally we can able to validate this structure. Okay, how to validate the structure, whether your modeled 3D structure of a protein, a target, or a receptor, whatever it is having a high quality or not. So to check the protein is of high quality, we have to go for validation. Okay, now let me proceed. Like actually, I thank again Professor Praveen Ramanujam, who is the coordinator of this FTP, who is the blue color shirt, and myself, I'm the course instructor. So as I previously mentioned, we have already in collaboration, we have completed nearly 6 certificate courses, and this is the 7th certificate course. I have delivered so many lectures in industry and also for Atal FTPs, and also BSME, IEEE, um, not only SERB, even AST sponsored FTPs. I have invited as a speaker to deliver talks on drug design and vaccine design in cancer. So this is my latest uh, project grant I'm working on that is related to drug design in hepatocellular carcinoma. And I also taking consultancy services from different institutions. One such institution, Sri Krishna Arts and Science College, some two MSc students have completed a project under my guidance. They also won first prize in Satyabama conference. A conference was conducted, and so they won a prize in that event. And even this one more faculty who has completed a part of a PhD under my guidance under consultancies. So myself, I'm working in working as a professor in Department of Biotech SVCE. So where we have excellent facilities not only to work on drug design and vaccine design, but also on drug and vaccine development. We have approved animal facilities, animal house facilities approved by CCSCA. Okay, so we are doing all this kind of experiments, and in the last 2 years, I also find my students has won the hackathon competitions in drug discovery hackathon. Under my guidance, 6 students have won 60,000 each as an internship stipend. So nearly they won 3.6 lakhs. Okay, I think they have received in year 2022. And also my PhD student, first PhD student, she has won this India Smart Protein Innovation Challenge. Only 20 members will be getting 1.07 lakhs. She is the topper of all the 20 people. So she also received the same 1.07 lakhs after winning phase 4. There are 4 phases. She has cleared up to the phase 4 and won the first prize. And related to one more first prize we had with the UG students on last year, which is related to competition related drug design in cancer. Okay. So these are some of the achievements from UG and PhD scholars. And now I can take you to the concept. I was explaining you something about target discovery, modeling, and validation, isn't it? So before going to the target, we should have a brief introduction on what is drug discovery. Okay, and you have heard so many terms like drug discovery, drug design, drug development. So what are the difference between these terminologies, these technical terms? Generally, for developing a drug, we should initially go for discovery, isn't it? So if you want to go for discovery, better to use the computational methods to reduce the time for discovery as well as to screen the compounds, screen the library. So it can be even peptide or it can be an antibody, whatever you're going to use as a drug, that has to be screened. And only the best candidate alone, you take it for preclinical followed by clinical trials. So only in preclinical and clinical, we call that as drug development process, isn't it? Even in the drug development, we have a type of technologies, like whether you're using nanotechnology or you're using a mitochondrial targeting. So however it is, if you want to check the drug testing process, obviously after developing the drug, you'll be testing in preclinical or clinical trials. Okay, so not the development has to link with the trials. Okay, now if you see this computational drug discovery, initially, I think this kind of a picture I fetched for 10 years before. So there they mentioned like for target discovery, they have taken nearly 3 years. But now, whether it took nearly 3 years, take an example of drug or vaccine design for SARS-CoV-2. We don't have a approved drug, but at least we had a kind of a vaccine that was not FDA approved, Covishield, Covaxin, not FDA approved, but still we are using the vaccines during the pandemic time, isn't it? So how much time they have taken, for a computational, for studying in computational part to come with a drug or vaccine at that time? They have taken just 2 to 3 months. Within 2 to 3 months, they came up with a kind of mRNA, they came up with a kind of a vectors, and they incorporated that. So all the kind of studies they completed within 2 to 3 months, not more than that. Even for screening the candidates, drug candidates, one scientist from California, I think California Institute of Technology. He used a supercomputer and he screened almost 2.1 billion compounds, okay, in virtual screening within 1.5 months. And he started publishing within 3 months. Now we have a high computational power just because of the rapid development in computer hardware, especially supercomputers. Just because of using the supercomputer with high performance computing, the drug discovery, the timeline is reduced gradually. Okay, now it is almost within 1 or 2 months they are coming with any kind of a drug or vaccine for any kind of diseases. Okay, that's most important to reduce the cost of development of a drugs. Okay, now if you see this kind of a computational methods, there are so many application areas, especially if you see the computation which is shown in the bottom. We have to think about the metabolism. If you are focusing on type 2 diabetes mellitus associated with a kind of a cancer, obviously we should have a knowledge on the metabolism, isn't it? The glucose metabolism. And if fat is accumulating more, then obviously even you have to think about the fatty acids, metabolism, all these things happening on, which can also lead to a kind of a cancer. Even the fats accumulation can lead to a type of a cancer. Then we should know what kind of metabolism. So here, what I mean metabolism modeling is we can able to design the pharmacokinetic parameters in the form of compartment models. Yesterday I was just terminated with one software called MATLAB. There is one commercial software called MATLAB, which can be used to study the pharmacokinetic properties. Okay, so if I go to the basics, then if I start explaining about the software and how to use, then you can understand. Okay, so we can understand what do you mean by pharmacokinetic in subsequent sessions. Okay, let me cover just the basic concepts around. So metabolism modeling, we can able to do it using the computational softwares, can be even MATLAB. Manufacturing modeling, protein structure prediction, we'll be discussing it on today. How to go for structure prediction, homology discovery. This is a kind of a structure prediction method. Pharmacokinetic modeling, of course, we have a separate kind of a heading itself called pharmacokinetic modeling, where we can study even for cancer. Okay, like in metabolism modeling, we can study about the T2DM, and if you want to study about the cancer associated with it, so we need both kind of knowledges, isn't it? And function prediction, because here now the patients always prefer a oral route of administration, and they want a single drug which can treat multiple diseases like a paracetamol, isn't it? The paracetamol, if I ask any person, even can it be a child, if I ask about what is a popular drug, they immediately tell about the name called paracetamol. What is paracetamol? It's just acetaminophen. Acetaminophen is the generic name, and paracetamol is the kind of a brand names, isn't it? So that kind of a drug is not only used for curing fever, but also can be used for curing. What else we can use? What are the benefits of using the paracetamol or acetaminophen? Can I ask you a question like this in the chat box? Can you type it? What is the significance of paracetamol or acetaminophen? One thing I told you, it is used for treating. This is for treating fever. What is the other significance? Fever is right. Good. Analgesic, cure headache. Very good. So you mean it's a painkiller. Good. So I got from Mohammed Rijwana. Good. That's what I want. I anticipated from you. I got the answers from your side. So we have two major kind of benefits, but we have a few more benefits also. Nowadays we are expecting a kind of a drug which can cure a multiple disorders, diseases, etc., etc. So obviously we have to link the types of methods and the type of application areas. Okay, whether the same drug can be used for metabolism modeling, and you can also check whether it can be used for pharmacokinetic modeling, can be cancer. So like this, we can try and we can even link the associations in the computational itself. Okay, and toxicology prediction. So even if you want to if you have in your computational studies, if you got a good result, okay, then you have to check whether a drug is having a toxic effects or not before going for in vivo. Even in vitro cell lines, your drug might not show any toxic effects in a particular concentration, but the same concentration, if you try to, or if you manipulate and if you try with the mice or rats, you find is more toxic. So obviously, even the toxicology prediction, we can also able to do with the computational softwares. So there are so many online websites available even to check with the mice or rats. Okay, so mostly there are many softwares to predict the toxicology prediction in rats in the online data and online tools itself. And drug synthesis model, even the drug can be synthesized in combinatorial ways in the computational itself. And then we can try to go for visual screening. Visual screening is one of the method to find the binding ability of a drug with a protein target. Okay, so we can synthesize the libraries and then we can try each and every drug, try to interact with a disease receptor, and we can check which is the best one. That we call it as virtual screening. Okay, so after drug synthesis modeling, we can go for virtual screening and disease simulation. If you want to simulate a disease, that is possible even with the softwares like MATLAB. Like what are the symptoms of what are the major organs affected, and these are all we can able to bring it. Okay, and also we have plenty of databases to show about the genes linked with the diseases. One gene can link with multiple diseases. Then what is the how about um, the gene interacting with the corresponding gene interacting with other genes? How about the interacting genes with connection with the same diseases? All these things we can able to check. Okay, if one gene is major responsible for disease, we should not limit up to that for going for precision medicine. We should also check whether the gene is also linked with a few more genes which can contribute, which can link with a particular disease what we are studying. Okay, all these things we have to go for disease simulation, product stability modeling. Okay, now we see one by one. First, start with the basics. What is drug design actually? Generally, in 1970s and 1980s, if any patient is suffering from any disease, obviously, we just take pestle and mortar. They take a kind of leaves or some kind of a roots, medicinal from the medicinal plants, and simply they grind it and they give it as a chorna, isn't it? So whatever they give as a chorna, whether it can give the promising results. So they initially try with the preclinical, whatever areas of medicine like Ayurveda, Siddha, Unani. There are so many branches of medicine, isn't it? So whatever they take from the natural sources, not only from the plants, it can be from the animals or microbial sources, algae, whatever they take. Okay, so they try to extract the compounds and they check with initially preclinical trials studies with small animals. Then they try with the clinical studies. But whatever they try, it gives a kind of trial and error based kind of a results. If you want to get a promising results, better go for structure based drug design. Okay, so in traditional based drug design, we are not majorly focusing on computational part, but if you focus on a computational part, which is a recent method, that is here we focus on each and every compound of the extract. So take a plant, if the plant is majorly responsible for curing a particular disease, then you should try to find which is the main compound which is curing that disease. Instead of taking the specific compound, if you're focusing on the all the plant compounds, maybe few plant compounds will aggravate the disease. We don't know which compounds can inhibit and which will aggravate. Okay, all these things we have to work it on. Okay, so all these things can be done through this structure-based drug design studies. So we should, for this, we should have a 3D structure of a ligand, that is a drug or inhibitor, and we should also have a 3D structure protein or a target receptor. Then only if you try to bind with the ligand with the target receptor, then we can understand whether based on the binding affinity, based on the energy, whether you have a good kind of a binding affinity or not. Okay. So this we can able to do only if you know the 3D structure of drug as well as the disease protein receptor.
Sir, voice is breaking.
Yes, participants, you can interact with me. Am I audible?
Yes, sir. Yes, sir.
Yeah, just now, isn't it? Just now? Yes,
Yes.
yes, yes. Okay, thank you. Yeah. We faced these kind of problems. Yeah. One thing. Also, I found I didn't interact well with you. I'm just taking you technology. So now I can ask. Can I have one or two interactions with you? Um, I just want to check. Okay, even the participants are many. How many are PhD scholars? So if you are PhD scholar, simply type yes. I want to check how many are PhD scholars. So I find many are UG scholars. Okay, only one, two, very few, three. Okay, very few are PhD scholars. Okay. Uh, how many are from MBBS or MD background? So if you are MBBS or MD background, kindly type yes. No medical students. No, while even when we are conducting FTPs or workshops, many MBBS and MD doctors join the kind of a workshops and FTPs. It's happening in my institution. So just I'm checking really are you from the medical background or not, because our theme is based on that. Okay, then I think majorly I can find how about faculties? Lastly, I want to check the teaching faculties. How many are from the teaching faculty background? How many are teaching faculties? I think very few. Only one or two are going to answer. Fewer are going to answer. Okay, maybe 10 to 20 will be there. Okay, good. Thank you. So thank you very much for this interaction, because I want to know whether you know the basics or not, because very few faculties are there and many are UG students. If my concepts are not understanding to you, because I thought you are from majorly from a faculty or a PhD scholars, so I'm taking in somewhat in advanced manner. If you're not understanding, halt me, break me, okay, and you can ask any doubts also. I think now I can go slowly for you. Okay, now I understand. So now if you see this drug discovery and development, okay, um, initially we can go for target discovery, then we can go for hit generation. The target discovery under is a disease protein information. Hit generation lead identification is something related to drug, a small molecule. A small molecule it can be a normal drug, a synthetic drug, a natural plant compound, or it can be even RNA, or even generally the lead identification on the other. But the ligand and etc., the ligand can also be an RNA or also a small protein. In antigen antibody interaction, if only if your antibody has to treat a specific pathogenic antigenic protein, then obviously the antibody is your major thing. Then under that case, okay, what antibody is specific to antigen, that is your lead antibody. Even I can tell that is a lead antibody, because we can generate so many antibodies. Even the structures of antibodies can be determined. But that antibody has to have more specificity with the antigen, isn't it? If a macromolecule, like an antibody, the antibody itself is a macromolecule, and the antigen is also a macromolecule. If this kind of a macromolecule has to bind with a macromolecule, it should have more specificity. If you have a key and lock, if only if a key is more designed to fit with the lock, then only it lock, isn't it? Similarly, the binding effect can be done. Okay, this is what I want to explain. And related to drug development, start with preclinical followed by clinical trials. So more about this, you can study in pharmaceutical textbooks like pharmacokinetics textbooks. Ramankar is one of the best textbooks. And if you see this major discoveries of drug discovery, so if you see the drug discovery, we can start with all this kind of cloning the new target, start with the DNA, RNA, whatever you want to work, and go for biological assays. Biological assays, here I mean, you can go for ELISA, you can go for PCR or RT-PCR, whatever you want to work. Okay, so we can work on the wet lab experiments, which we call it as a biological assay. Screening, we can screen if you have multiple antibodies, or if you have multiple drugs, you want to check which drug is best for a particular protein, then obviously you can screen accordingly. Then we can also go for a computational part. We can do with the computer aided drug design using the rational design or medicinal chemistry. And drug development. In drug development, we can able to test the drug by checking the dosage of a by checking a dosage of a drug by in vitro studies, or even through majorly through in vivo studies. Okay, so before going to the in vivo studies, the scientists, anyone, even from the CCSC committee members, they ask you to do first initially computational studies, later in vitro studies, then they will be asking you to go for in vivo. Okay, then if I ask me, then obviously after the computation, we have to come with the in vitro, then we have to go with the in vivo. And during the in vivo experiments, you can also formulate it using various technologies. You can formulate the drug using various technologies and various ingredients. And then you can also check finally with the small animals once again. Then you can go with the clinical trials from 1 to 4 clinical trials. And finally, the drug elucidation. The drug elucidation can also be checked with model organisms. Okay, with model organisms, we can check with the zebra fish. We can work with the nematodes. So whatever we are going to work, okay, we can use a model organisms and we can study the drug dosage response, which comes under pharmacodynamics. Here, if you study the pharmacokinetics, so ADME is something related to pharmacokinetics, and you can also study the pharmacodynamics in the drug elucidation. And if you find any because of genetic effects, if you find a disease is originated, even we can study the genetics later. So initially, if you want to study the disease, you have to study the disease in the development. And finally, if the disease is caused even because of mutations, then for the mutated kind of gene expression for a particular disease, you have to work separately, exclusively, and then you have to discover the new drugs for that, new drugs and new targets for that. Got it? So target identification, there are so much description on that. If you see the overall drug targets, okay, the major drug targets are the receptors. Okay, and next to the drug receptors, I mean surface receptors in the diseased protein. I mean, in the diseased cell. In the diseased cell, we have so many surface receptors. These receptors are important targets. Okay, and if you can design the drugs or whatever the antibodies, you can try to bind and you can check the affinity. So next to the receptors, you can go for enzymes. So nearly 28% of enzymes are important even for the diagnosis. If you say diagnosis or prognosis, the disease is detected only through the enzymes, isn't it? The biochemical parameter of a specific disease, if you want to identify, if you want to diagnose, obviously we should use the enzymes, isn't it? So second most important target are the enzymes after the receptors. Then hormones, of course, these are also important. We also have few more target receptors. Actually, there is one concept called target discovery. That's what I was discussing from the beginning. There is also one more technical term called target deconvolution. What do you mean by target deconvolution? If you see the target deconvolution in the second line, you can see in this scenario, you begin with a drug that appears efficacious and the target is identified retrospectively. Generally, for target discovery, what you do is you are not going to start with a drug and you check with whether a drug is binding with what type of receptors. You understand? In target deconvolution, you start with a single drug and you try to check whether the drug is binding with what kind of receptors. Generally, we won't go for target deconvolution. We generally go for first identifying a target, okay, and then you screen with multiple drugs. We have to check, like, if you have a disease protein and the diseased protein, அதாவது ஒரு cell, for example, you can tell me any disease, can be if you take a liver cancer. If you take a liver cancer, if you want to treat a liver cancer, first you should start with what is the major protein. Okay, what is a major protein or target or receptor which is important for treatment of that liver cancer. So first one, though, you have to identify a protein, because even in the feedback I found a few participants have mentioned also use Tanglish. Always you are speaking in English. If you speak in the, I think even the faculty has mentioned this kind of a feedback. I have studied that also. Okay, if you use a kind of a Tanglish, then even it will be better understanding for the other participants. Okay, so even I can interact with it and then I can complete this explanation on convolution and discovery. How many want Tanglish? You want me to stick strictly English or Tanglish? 1, 2, and if you're not dominant, okay, what to make? Many are going for English. See, the English is dominating, obviously. Okay, so I try to reduce as much as possible to avoid Tamil. But okay, for few I can bring it Tamil. Okay, now if you see this kind of technical terms, generally if you identify a disease, you have to identify a protein target. Okay, if you identify a protein target, for the protein target, you come up with so many kind of drugs and you screen it. That we call it as a target discovery. But this is something natural. We are not following exactly. That is, you are using as one drug and you check whether the drug is treating a drug is binding with a different receptors of different diseases. For example, if you have a paracetamol, whether it's if it is curing fever, I can start with the paracetamol itself because many of them don't know about the mechanism of action of acetaminophen or paracetamol. Can someone tell me what is the major target receptor for curing a normal fever, not a bacterial fever, a normal fever? What is a major target receptor for curing fever? You are taking acetaminophen, isn't it? Good. You tried with blood cells. Of course, the blood, which is giving a high rise in temperature, a slight high rise in temperature. We have nearly 36.7 degrees centigrades, a normal temperature in our human blood, isn't it? In Fahrenheit, we get 98.6 is the average temperature. In degree centigrade, 36.7. But if you come across 37.2, 37.3, that itself we call it as a fever. That is, if you're crossing 99 degree Fahrenheit, we call it as a fever, a slight increase in temperature, isn't it? But my question is, even if you're targeting a blood, you're not targeting all the proteins in the blood cells. If you take a blood cell, you have lymphocytes, monocytes, mast cells, dendritic cells, macrophages. So many cells are there. Okay, eosinophils, neutrophils. So many cells are there. But whether we can, the drug, whatever paracetamol you take, it can be a 500 mg or 650 mg. If your weight is 75 and above, generally the doctors prescribe to take 650 mg, or else they ask you to take just 500 mg. You accept that? But my question is, what is the major target receptor your acetaminophen or paracetamol is going to target to reduce fever? No, I didn't get result. Still, PGE2, okay, but that's not a major target receptor. What else? Can I go with an answer? COX-2. Can someone expand this? COX-2. What is COX-2? To remove the pyrogens in the blood, we need this kind of a drug. Okay. Of course, if you take a drug, acetaminophen, it's going to bind with this kind of it. Very good. The answer is cyclooxygenase. Okay. So this is the target receptor for this is an antipyretic, which is the acetaminophen, which target this kind of a receptor. You see the wonder. What kind of wonder we can see in the paracetamol? The paracetamol, you take just a 500 mg, and this 500 mg is going to target 6 liters of blood. Wherever you find the COX-2, it just has to bind with it. And you know, what is the residence time of a paracetamol? Can someone answer me? Because this is most important. Okay, just you should also know about dosage, and you should also know about the residence distribution of paracetamol. If you take a paracetamol, how much time the drug is going to reside in the blood? We know we are going to find a drug protein binding happening inside our body, isn't it? What is the residence time distribution of paracetamol? Can someone tell me the correct one? Correct answer? 6 hours. Good. Okay, not bad. So 3 to 4 hours. Oh, madam. Stay fine. 1 hour. Ayyo. Wow. If you are taking 24 tablets in one day, the person will be dying soon. Very good. The answer I got from Lalita Devi. Yes, you are right. 8 hours. No. I don't want any answers. I already told you the answers. The residence time distribution, I mean the drug protein binding is exactly 8 hours. So only we are taking 3 doses a day. If you take even 2 doses per day, it's not recommended by doctors. You are taking your own decision. Definitely, this kind of treatment won't help you uh, to get relief from the fever. Nor you can relieve from the fever. Even if you have a body ache, it will be increased tremendously. So you should have, you know about the residence time distribution of any drug in our body. So for this, of course, pharmacodynamic studies is important. I was explaining you something about drug elucidation, isn't it? So the in drug elucidation, after the drug development in pharmacokinetics, the pharmacodynamic give you a better understanding on that. Okay. Now I was explaining about target discovery and target deconvolution, or the two different methods we can use to bring to discover a drug, okay, for treating a particular disease. Okay, this is some theoretical information. If you see this kind of a target discovery, okay, even if you have a single protein target, if your drug is going to target a single protein, we can't certain that it can able to cure the disease, especially for cancer. For cancer, might be originated because of few genes, not just a single protein is important for raising the cancer. A few proteins, that is, if even if you identify a major protein, that major protein is a hub protein which is going to interact with other proteins. So you have to go for the binding affinity even with the interacting proteins of that hub protein. For this, you should have a knowledge on network pharmacology. Okay. So more about the network pharmacology we'll be seeing in subsequent sessions. So I was even yesterday I was explaining you about stage string DB. Understand? So because I was explaining you about cancer, okay, in cancer you can't be treated like a paracetamol. Okay, for the curing fever. Paracetamol, we generally use it for curing fever, not only for bacterial, and even for viral origin, generally the doctors prescribe paracetamol, isn't it? For safety, they have an idea, even along with the virus, even if you have a bacterial kind of a pathogens inside, then obviously they also give, also sometimes they also give paracetamol, okay? But generally we take antivirals like wherever it end with vir, retinavir. So whatever we call it as vir, we call that as antivirals. So we have to, the doctors have to generally prescribing this kind of antivirals for treating the viral fevers instead of relying on paracetamol, which is going to target only the bacterial origin. Okay. Okay, now we can go across. This network pharmacology will be studying more about in subsequent sessions. Now let me complete about the target, where we can find the target receptors and how to model the proteins. So if you see this website called Phosphocyte Plus, we can able to take the diseased information, protein information also we can get. We can get also the cell line tissue. There is one website called cellsignaling.com. Okay, so where you can find, you can download the pathways, you can download the markers for a particular disease. Markers, I mean target proteins, microglial markers. Okay, so something related to neurons in brain. Okay, so we can go across, and if you go for nephronic disease, we go for nephron. So all these things we can able to find. But generally here they majorly have this kind of research, and even we have antibodies and agents for the SARS-CoV-2. I think this I have already taken string DB, where you can find protein-protein interactions. And if you want to have information on a protein-protein interaction kind of information, you have to type the PDB ID of a hub protein. If you click that, you can able to find what are the proteins interacting with this subprotein. Okay, even that kind of information. Not all the proteins, we can find one or two proteins interacting with with this other proteins. So I can tell you that when I take you to the website, click pathway to explore more on the pathways, to more on the drugs, drug targets, all these things. You can use the cake pathway. For this, the website URL is genome.jp/cake/pathway.html. Stage, I told you, drug and protein interactions. There is one more database called small molecule pathway database. So here, if you click, if you go to this URL, smpdb.ca, California hosted this website. So you can, if you simply go type a small molecule, it connects you with the molecule connects with what kind of um, proteins. So all this information you can find here in this small molecule pathway database. For a drug, we can find the interconnecting pathways. And if you see the next database, therapeutic target database. So the URL is http://db.idrb. So the URL is there. Now in the therapeutic target database, you have to simply type the protein name, protein target. If you click search, obviously get more information, structure, sequence, all this information are retrieved. The next we go for biomine.cs.vcu.edu. So in this, you can find protein drug interactions. If you type a drug, if you click search, you will be getting the drug binding with so many protein receptors. Okay, astagalomide, I found nearly it's having an interaction with this much number of proteins. If you click search, you can get more information on that. Therapeutic drug target database. Here, you have to simply search for a drug, type a drug name, then you can able to find the drug is binding with what kind of receptors. Instead of finding the target receptors, if you want to find a drug, better click this target search, type a protein, then you can get some kind of information on the target search. Instead of drugs, we'll be getting the proteins. Intact molecular interaction database. This is from ebi.ac.uk. So we also have already prepared COVID-19 dataset. You can try the example. MINT database, the molecular interaction database. You can type a gene or a protein name and you search it, obviously get this information. Not only you'll be getting the information on that, we also get information on the wet lab techniques used to post the gene information on that. In details, you may get the expression of that and molecular modeling. So, so from up to this, I was expanding about a different databases. Now I can take you to the websites where you can find a 3D structure of proteins. If you want to find a 3D structure of proteins, better go to the Google and you can initially type for RCSP/PDB. Even if you type RCSP/PDB, I mean spacebar PDB or simply PDB, it takes you to the same website called Protein Data Bank. PDB stands for Protein Data Bank, RCSP, Research Center for Structural Biology. So this is the major web resource where you can find, the web resource where you can find major 3D structure of proteins. So maximum of 2.14 lakh structures are available in this web resource. Okay. If you want to do any research related to drug design, vaccine design in computational, we start taking the structure of a protein from this website defaultly. Do you know any other databases for 3D structure of proteins? So I was explaining you that RCSP PDB is one of the repository for downloading 3D structure of proteins. Do you know any other databases other than PDB? This is my question. Interact with me. Type your answer in the chat box. What are the other databases used to download 3D structure of proteins other than PDB? Because I want to check how much knowledge you have. I have to keep pace with you, how much you understand. Uniprot, generally we can find more information on the secondary structures. Okay, you can link with the cath, SCOP, etc. Alpha fold cut. What else? When you plot, we get the secondary structures. I mean, it's a secondary database. We can find more information secondary database. Link with the PDB, obviously. What else? ProSite? No. Anyone know? We also have a 3D structure protein database in NCBI. What is the in NCBI? What is the 3D structure protein database? We have so many databases in NCBI, isn't it? The final year might have studied bioinformatics. They know we have GenBank, GenWeb, Blast is a tool. Okay, we have so many databases. Even genome we have in NCBI. But my question is, what is the 3D structure protein? So I answer at present, molecular modeling database. Okay, so where to find the molecular modeling database? Simply go to the NCBI website, National Center for Biotechnology Information. That is the expansion, NLM, National Library of Medicine, NIH, National Institute of Health, GOV for US content. If you want to download a 3D structure, you go to the structure and you type either the PDB ID. The PDB ID is generally 4 letters. It's a 4-letter. It's generally a alpha numeric. Okay, it's a combination of an alpha numeric characters. If you know the protein name, type the protein name. If you're focusing on insulin, type insulin. If you want to type a myoglobin, hemoglobin, whatever protein you type it. And if you click, obviously you get an information. Now, presently, I am typing a random kind of a PDB ID. Now, I am getting, see, the 3D structure information on this. What is 1XIH? You can download the 3D structure. Okay, the same, we can able to make it up even in this website. Okay, that is in PDB website. Go for typing a protein name, or you can type the PDB ID. Okay, so you can find the same information. That is, this is something related to D-xylose isomerase. You're also getting the same, isn't it? Okay, so in this, if you want to download, click download files. Better go to this, the download files, and download as PDB format. If you click this PDB format, you can download wherever you want to make it out. Now I'm presently saving it in desktop. Now I saved that protein file in 3D format. Okay, so now whatever file I downloaded, either from RCSP PDB or MMPDB, this you can visualize it using any visualizer. I have already installed a visualizer called BioVia Discovery Studio, which is a free visualizer. Okay, anyone can download from this website. BioVia Discovery Studio 2021, very user friendly. So whatever structure I download is a xylose isomerase. Okay, the 3D structure of a protein looks like this. The red color, the ribbon kind. Okay, this is actually alpha helices. In this is actually a secondary structure of a protein. Okay, this red color helices, we call it as alpha helices, and the blue color we call it as a beta strands. Alpha helices and beta strands are major secondary structures of a protein. So whatever you can able to see a 3D structure of a protein, if you want to edit the coordinates, that you can edit using WordPad or Windows Write. Don't use any other editors for editing the coordinates. Whatever 3D structure we found, the coordinates of the PDB file are present in text form, which has to be opened one in WordPad or the Windows site to edit any content inside. Okay, so what are the information we can find in a protein structure? We can able to find in a header, we can find the major title of a protein, and in title you can find the exact name of the protein. The exact name is enzyme D-xylose. Okay, so that it's not as a xylose is not an enzyme. So actually xylose isomer is something like that. Okay, so that's what I can able to find. D-xylose, yeah, here you can find isomerase, D-xylose isomerase. This is the protein name. And if you want to find the organism, okay, the organism is Streptomyces rubinoides. And if it is genetically engineered, you can also get the information whether it's engineered or not. Okay, and related what kind of wet lab technique is used to get this 3D structure. Here it is mentioned as X-ray diffraction. So like this, we have so many parameters we can able to see here in the home page itself. See, this protein is deposited in 1994. Based on that, the authors who deposited the resolution at that time itself, they came up with 1.7 angstrom. If you want to find whether a protein is good or bad, okay, based on the resolution, we can also able to decide. Okay, if you have 2 angstrom and less than 2 angstrom, whatever drug design or vaccine design you're going to take with this, you're going to study with this protein, will give a precise result. 2 angstrom and less than 2 angstrom is a high resolution protein. Okay, so this is how we take a protein receptor. We can visualize a protein receptor using the visualizer and read the coordinates using the Windows right or WordPad. So more information you can search like this. Okay, if you have a structure, okay, no problem. But if you don't have a structure, how to go for modeling? For this, we can go for homology modeling, one of the method using Swiss modeler. Swiss modeler is one of the method to model the proteins. Okay, so for this, we should paste the sequence and then we have to simply click build model. So if you type a project title and or email or the optional things, if it takes a minimum of half an hour to generate the result. But if you have typed the email and the project title, obviously it generates the results and send it to your email ID. So to model the 3D structure, obviously we can use this kind of a universal kind of a tool which uses a homology modeling method for protein structure prediction for unknown structures. Got it? So this is something about the target modeling. And if you want to go for validation, obviously you can able to go for assessment of a protein using Ramachandran plot and other methods. Okay, there is a lot to describe about the molecular modeling. Okay, so I just take few concepts around because I started 5 minutes late. So with your permission, I can take 5 more minutes. So if you go for a target protein structure prediction, if you don't have a structures, we have just 2.15 lakh structures in PDB website, isn't it? But if you see the database in NCBA itself, we have a protein sequence database. Where is NCBA? If I take you to the NCBA, okay, instead of going to the structure, you go to the structure and if you type a protein name or a PDB ID, you'll be getting the protein information. But if you want to find a protein sequence, you can click protein. If you click a protein and if you go for typing the name of a protein, okay, it takes you to the specific protein sequence alone. Okay, then this kind of a database is a GenPept. You know, the name of a database in NCBA is a GenPept for a sequence. If it is a structure, it is MMDB. Okay, but if you see the protein sequence databases, you have more than 40 million sequences, 40 million 400 lakh structures, 400 lakh sequences are there in NCBA. You have more than 400 lakh sequences. Okay, but you have only 1.5 lakh structures, 1.2 lakh structures. Then if you don't have a structure, how you can able to discover a drug for a particular disease? So only I'm telling you to go for modeling of a protein structure using the sequence alone. Sequence or using the sequence itself, we can able to determine the structure. For this, we have 3 methods. One is homology modeling, number 2 threading, the third is ab initio. So from XPASI, we have so many tools to work on this online tools for building the 3D structure of proteins. Each and every tool uses unique kind of a machine learning technique pattern to come up with a structure. Okay, then if you go to this Swiss modeler, here I am just pasting the sequence I told you. You can feed this as an option like a project title in the email if you want to generate the results in your mail. Click just if you click this search for templates, it take you to this page where you can also able to select your template based on your requirement. If you want a kind of a more identity, or if you want to give more priority for the resolution, even you can select, instead of first checkbox, you can select only one. Only one template has to be selected for a sequence. Okay, select only one thing and you click build model. If your binding site, if your drug is not binding with the binding site, then you can select the other kind of a thing and you also build models on that, and then you can try with it. So generally in steps in homology modeling, if it simply click, you will be getting a structure, but these are the steps happening inside virtually, okay, to develop a kind of a 3D structure. Finally, after developing a 3D structure of a protein, you can download the structure like however I downloaded in the desktop. You can visualize it using the Discovery Studio or RASMOL or whatever you have mentioned there. Okay, so you can open the protein structures in any protein visualizers. But there is one thing called validation of the protein. That is, if you want to check whether a protein quality is good or not, you can able to check by assessing the stereochemical quality of a protein using Ramachandran plot. In Ramachandran plot, we can able to find in the first line as a residues in most favored regions. The residues in most favored regions should be more than 80%. Okay, if only if you have 80% residues, then more 80 or more than 80, then only you can take the protein structure further for docking, simulation, etc. Okay, the assessment has to be done by checking the stereochemical quality using ProCheck tool, where it generates a Ramachandran plot. There's so much kind of a protein science to know about how the stereochemical assessment is done. Also, I can take you that even you can use iTaser, which uses a threading concept. Just simply paste your sequence, paste your email ID, and then you just click submit. Obviously, get a 3D structure. This is a flow, what is happening inside. And we can also predict peptides using the PepFold3 server. So more about this we'll be seeing in the subsequent sessions. Whatever missed out in this session, we'll be discussing it in the continuing sessions, okay, in the remaining 12 more sessions. Any doubts up to present? If you have doubts, you can stay for 1 or 2 minutes. The remaining participants can take. So thank you very much for the patience and interaction. Any doubts you have, participants, at present, you can type in the chat box. And I request you to come and join at on or before 7 o'clock. Don't join in between, because even it creates a disturbance to the others, okay, including me. Okay, even you are missing something. In the initially, I go for recapping of what we discussed in the last sessions, previous sessions. You may miss that also. Okay, so join earlier. So thank you. Try all this, okay, and I believe even your coordinator will be available from tomorrow onwards, and he also going to share you the recorded videos from tomorrow onwards. Thank you very much. Yeah, he take care of all this. Swastika. Have a great day to you. Bye. Good night.